Hey everyone, this is the exam 4 review from our textbook, Biology, Concepts, and Investigations by Hoefnagels, covering chapters 9 and 10. Let's go ahead and get started.
Dr. D, 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 Dr. D. Explain stuff.
All right, let's get started with exam 4 review covering chapters 9 and 10, starting with chapter 9, Sexual Reproduction and Meiosis. One thing you should know for sure is that asexual reproduction is when a parent replicates its DNA and splits into two, and this results in two genetically identical offspring. And remember, in asexual reproduction, this can occur in prokaryotes by binary fission and even in single cell eukaryotes by mitosis. So, for instance, if this is, let's pretend this is an amoeba, if this amoeba splits into two by mitosis, that's how an amoeba would asexually divide, right? Resulting in two genetically identical cells. So you should know that mitosis and binary fission are ways that single cell organisms can reproduce asexually. For sexual reproduction, we must have meiosis, which results in genetically diverse gametes, the sperm and the egg, and those come together to form the fertilized egg or zygote. So, again, you should know that bacteria divide asexually by binary fission, and single cell eukaryotes, they also divide asexually by mitosis. And remember, in sexual reproduction, the offspring are genetically diverse, not genetically identical. And so you should know that the main benefit of sexual reproduction is genetic diversity, which allows organisms to carry on in the face of selection in nature. And you should definitely know the terms diploid and haploid. Do you recall that diploid refers to organisms that have two homologous sets of chromosomes, one set from the egg, one set from the sperm, whereas haploid refers to only one set of chromosomes. So, for example, bacteria are all haploid. Uh, Sperm are haploid. They only have one set of chromosomes, 1 through 23. Eggs are haploid. But when the sperm fertilizes the egg, that's diploid, right? Because now you have your two homologous sets of chromosomes. So in that case, a zygote, which is a fertilized egg, is a diploid cell, right? You should definitely know this, the difference between homologous chromosomes and sister chromatids. Remember that homologous chromosomes are not identical. Each comes from a different parent. So one homolog comes from the maternal source, from the egg, and one homolog comes from the paternal sperm. And when the sperm fertilizes the egg, that's when you have both homologs. Remember that homologs share the same chromosome number, like chromosome 1, chromosome 2, or chromosome 3, for instance, but they don't have the exact same genetics, right? They have the same genes on those chromosomes, but not the same exact alleles per se. So you should also definitely know the difference between a gene, like eye color, and alleles, which include, for instance, versions of eye color, brown allele, blue allele, green allele, right? So gene versus allele. Homologous chromosomes possess the same genes, but not necessarily the same alleles because you inherited one homolog from mom and one homolog from dad. Whereas, do you remember sister chromatids, on the other hand, are identical? They exist after S phase of interphase. Do you remember S phase of interphase, synthesis phase, where all the DNA is copied? when all the DNA is copied during S phase of interphase, that's when sister chromatid pairs form, and that's when you have two copies of every single chromosome. So, for instance, a cell that otherwise has 46 chromosomes now has 92 because you've copied each and every chromosome. So, sister chromatids are identical in every way, but homologous chromosomes, they share the same chromosome number and the same types of genes, but one was inherited from mom and one from dad. So they are not identical. So do you remember clicker question number 2? The image below shows 2 chromosomes from one cell. Which statement is true? A, both came from the person's mother. No, that's not true, right? Because this chromosome came from the egg and this one came from the sperm. B, the 1 and 2 pair is identical to the 3-4 pair. No, the 1 and 2 pair is identical to each other. The 3-4 pair are identical to each other, but 1 and 2 is homologous to 3 and 4. So it can't be B. C, 1 is identical to 2. That's correct, isn't it? 1 is identical to 2 because these are sister chromatid pairs. And let's see why D is wrong. D is wrong because the chromosomes have not been replicated yet. They have been replicated. That's why you have sisters. So the answer has to be C. And again, you should definitely know the terms haploid and diploid. Haploid referring to only one set of chromosomes, whereas diploid referring to two homologous sets of chromosomes. Sperm are haploid, egg are haploid, and bacteria are haploid as well. But the zygote is diploid, and animals are diploid, plants are diploid. And again, the zygote refers to the fertilized egg, and it is definitely diploid. One thing you should definitely know too is that meiosis results in 4 cells, right? Because meiosis undergoes 2 rounds of cell division, right? Remember, uh, a germ cell divides by meiosis 1 to result in 2 cells, and then each of those divides again during meiosis 2 to result in 4 gamete cells. So you should know that 4 gametes result at the end of meiosis. And you should know that those 4 are haploid, and you should know that those 4 are all genetically different from one another, not genetically identical. And so, what is the purpose of meiosis? The purpose of meiosis is to produce the haploid gametes, right? The haploid gametes meaning the sperm and the eggs, which are totally genetically shuffled around, right? Each one is genetically different than the next. Do you also remember what happens during prophase 1, prophase 2, what happens during metaphase 1 or metaphase 2? Do you remember all that? Let's see if you can beat Wicket, okay? Let's see if you can beat Wicket. Where do the tetrads form and crossing over happens? Can you beat Wicket? That's right. Wicket is he's always right, you guys, as you know by now. Yes, he said during prophase 1. During prophase 1, that's when tetrads form and crossing over happens. And remember the purpose of crossing over? To increase genetic variability, increase genetic diversity. What if I asked you this? During which phase of meiosis does independent assortment occur? Remember what that means? That means the tetrads lining up along the metaphase plate down the center of the cell randomly. Where does that happen? Can you beat Wicket? That's right again, Wicket. That's right, metaphase 1. So you should know that independent assortment occurs during metaphase 1. And independent assortment is also responsible for genetic diversity during meiosis. So those are the 2 big places where genetic diversity are made. One is crossing over, 2 is independent assortment, And then the third one's actually the fact that a random sperm fertilizes a random egg. Those are actually the three sources of genetic variability during the process of meiosis and sexual fertilization. Do you also remember that during meiosis begins with diploid germ cells that exist inside of the ovaries and testes? So it's the diploid germ cell that undergoes meiosis 1 followed by meiosis 2 to produce the haploid sperm and eggs. Now for clicker question number 3, and don't forget, these clicker questions may appear on the exam, so it's worth remembering them and learning them. Clicker question number 3, a zygote of a ferret contains 40 chromosomes, not the 46 like you and me. How many chromosomes are in a ferret's sperm cell? Remember, if the zygote is diploid, so the zygote is going to have two homologous sets, right? So if the zygote is diploid and it has 40 chromosomes, that means it received 20 chromosomes from the sperm, 20 chromosomes from the egg. Does that make sense? So the answer should be B, 20. Another concept you should brush up on for the exam is this concept of monozygotic twins versus dizygotic twins. Remember, monozygotic twins are identical. This is because they come from one zygote. That's what monozygotic means, one zygote. So one sperm fertilized one egg to produce one zygote. However, at some point, this resulted in two offspring, right? Two embryos. So you should realize that monozygotic twins are genetically identical because they came from one sperm and one egg total to make one zygote. Dizygotic twins, on the other hand, you should know that this is what's known as fraternal twins. This is where the offspring are not genetically identical, and this is because two different sperm fertilized two different eggs in the same womb at the same time, right? And this resulted in technically twins, but not genetically identical twins because they didn't come from the same sperm and the same egg. And remember, no two sperm and no two egg have the same genetic makeup. So this leads to clicker question number 4. Many sets, and again, you may want to pay attention to these. They may appear on the exam. Many sets of triplets consist of a pair and a spare. That is, two identical babies and one non-identical baby. So triplets, two are identical, one is not. Producing this arrangement requires how many sperm and how many eggs? Well, think about it. Identical twins come from one zygote, right? So that means one sperm and one egg. And then the other twin, the fraternal one, must come from a different sperm and a different egg. So the answer has to be what? 2 and 2, right? Two sperm and two eggs, resulting in 3 people, right? So I hope that makes sense. Next, you should remember that errors can sometimes occur during meiosis. And the most common error is called non-disjunction. This is where the chromosomes don't separate properly during either anaphase 1 or anaphase 2 of meiosis. And do you remember that this non-disjunction is responsible for genetic disorders such as Down syndrome, which is called trisomy 21, as well as triple X syndrome, Klinefelter syndrome, Jacobs syndrome, and Turner syndrome. You should realize that these are all syndromes due to non-disjunction. So, clicker question number 5. In Turner syndrome, a female is born with only one X chromosome. This disorder could arise as a result of non-disjunction in A, sperm formation, B, egg formation, or C, either sperm or egg formation. It should be C, right? It could occur in either the sperm or the egg. All right, and that leads us to the end of chapter 9. I think I touched on everything that was important. One more thing before I forget. How many, can you beat Wicket? How many alleles does each individual have for each gene? That's right, Wicket. Two alleles. You inherited one allele, let's say for eye color, you inherited blue allele from mom and brown allele from dad. So each person has two alleles for every gene. Okay, so remember that. But I think we touched on everything important for chapter 9 for exam 4. So let's take a quick little break time with Gizmo and Wicket, see what these little guys are up to. We'll be right back for chapter 10 to wrap up this review. All right, welcome back from break time with Gizmo and Wicket. Let's carry on with chapter 10, Patterns of Inheritance. Remember that a locus is a specific location on a chromosome in which a gene exists. Again, remember the terms homologous pair of chromosomes, which are not identical because each is from a different parent, versus sister chromatid pairs, which are identical because they are copies of one another. Also, remember Mendel's important experiments, his monohybrid cross versus his dihybrid cross. Remember that true breeders, Mendel started with true breeders for every cross. Remember that true breeders always look like the parent when self-fertilized, whereas non-true breeders can include organisms or offspring that don't look like the parent. Do you remember the dominant and the recessive traits? In the F1 generation, the dominant trait was expressed. The dominant trait masks the recessive trait. Also, remember the terms genotype versus phenotype. With genotype refers to the alleles that an organism possesses. For example, big Y, big Y, big Y, little y, little y, little y. These are genotypes. Whereas phenotypes are what we see, yellow versus green. And you should definitely know this for the exam. What Mendel would call a true breeder, we know now as homozygous. So, for instance, if it was a yellow true breeder, and if yellow was the dominant allele, then yellow is the dominant trait, and the genotype would be big Y, big Y. Big Y, big Y is the genotype for the phenotype yellow, and it's also the genotype for the true breeding dominant plant. Whereas little y is the genotype for the green pea plant, and it is the homozygous recessive organism. And what Mendel discovered was that non-true breeders are actually heterozygotes, which means they have one dominant and one recessive allele. So again, true breeders have two of the same allele, either big Y, or little y, and non-true breeders have two different alleles, big Y, little y. So true breeders are homozygous and non-true breeders are heterozygous. And remember, in a monohybrid cross, Mendel crossed a true breeder yellow pea with a true breeder green pea plant. You always have to start with true breeders in a monohybrid cross. Also, do you remember from lecture what was the phenotypic ratio of a monohybrid cross? Can you beat Wicket? That's right, Wicket. The phenotypic ratio is 3 to 1. 3 dominant phenotypes to 1 recessive. So, for instance, 3 yellow peas for every green pea, or 3 purple flowers for every 1 white flower. Also, do you remember, can you beat Wicket? What was the genotypic ratio of a monohybrid cross? Can you beat Wicket? That's right, Wicket. The genotypic ratio of a monohybrid cross is 1 to 2 to 1. 1 homozygous dominant, so for instance, big Y, big Y. 2 heterozygotes, which is big Y, little y. And only 1 homozygous recessive, little y, little y. So you should know for this for the exam for sure. In a monohybrid cross, the phenotypic ratio of the F2 generation, the phenotypic ratio is 3 to 1, whereas the genotypic ratio is 1 to 2 to 1. You should remember that for the exam. And remember, what Mendel would have called true breeders, we now know as homozygotes. What Mendel would have called a non-true breeder, we now know as heterozygotes. All right, this leads us to clicker question number 1. Do you remember cystic fibrosis is caused by a recessive allele, little f? If a healthy carrier and an affected individual have a child, what is the chance that the child will be affected? So remember, a healthy carrier would be big F, little f, and this would be crossed with an affected person. This would be a little f, little f. Remember what you need to do a Punnett square? You need to figure out the genotypes of the parents, and you already know the genotypes of the parents. One parent is big F, little f. The other parent is little f, little f. This means that we can determine the gamete types. That's step 2. Step 2, that we determine that the one parent, so let's say the sperm could be little f or big F. The other parent, let's say the egg, could be only little f. We place the sperm information, the types of sperm on one side of a square, and the types of egg on the other type of the square, other side of the square, and we see that there are only 2 outcomes possible: big F, and little f, little f. Half of the time you're going to have big F, little f. Half of the time you're going to have little f, little f. So the answer should be what? Can you beat Wicket? That's right, Wicket. 1/2. The answer should be C. 1/2. And before we move on, let me ask you this. If Mendel crossed a true breeder yellow plant with a true breeder green plant, and he only saw yellow offspring, what would that tell you about the yellow allele? Can you beat Wicket? This is an important concept for the exam. That's right, Wicket. Yellow is the dominant allele. That would tell you that yellow is the dominant allele because that F1 offspring would have a yellow allele from one parent and a green allele from the other parent, so it would have a big Y and a little y. And if all of the offspring are yellow, that tells you that yellow masks green and yellow is dominant to green. Now let's talk about the dihybrid cross. Do you remember that Mendel did the dihybrid cross to look at 2 traits at the same time? And remember that Mendel showed that different traits are not linked. You should know that during the dihybrid cross, Mendel showed that different traits assort independently of one another. And so this shows that independent assortment occurs, that different traits assort independently of one another. That's an important point about the dihybrid cross. Also, do you remember with the dihybrid cross what types of sperm and egg can be made? So you should know that when we're looking at a dihybrid cross where you're looking at two completely different traits. So for instance, here if you have a little r and a little y, then the sperm and the egg, or let's say the eggs can have little r and little y information, right? Because little r has to do with shape and little y has to do with color, right? So you have little r, little y. You can have a little r and a little y inside of an egg. You can also have an r and a y inside of the sperm because r and y are two completely different genes. R has to do with shape, Y has to do with color. But what can you not have? You cannot have two Ys or two Rs inside of a gamete unless it's due to non disjunction, which would not be normal. Does that make sense? So gametes can have one of each type of gene, like a little r and a little y, but they shouldn't have two of the same type of gene. So two Ys or two Rs. So remember that for the exam. And so if I asked you, what did Mendel show with his dihybrid cross experiment? What would you say? Remember, it was the law of independent assortment that different traits assort independently of one another. So different genes assort independently of one another. And remember, this was a callback to independent assortment during metaphase 1 of meiosis, because that's what explains Mendel's observations. During metaphase 1, remember how those tetrads lined up independently of one another? That explains how independent assortment works in Mendel's experiments, how different traits or different genes assort independently of one another. All right, this leads us to clicker question number 2. Why doesn't the Punnett square below include a female gamete with the genotype RR? So look at this. Why does the female gamete have the genotype RY and RY? Why not RR? Do you remember? It's because RR would be diploid, right? You could only have one of each gene. So one R and one Y is acceptable. But the only reason a gamete would have two R's is if it had non-disjunction. Do you remember that? And that's not normal. It's abnormal. So let's see what answer choice explains that. A, each germ cell only has one R allele. Now that doesn't make sense because remember, germ cells are diploid. Germ cells have two R alleles. See, this is the germ cell which has two R alleles. B, the Y allele is dominant to the R. That doesn't make sense. That wouldn't answer your question. Let's look at C. The R alleles separate during meiosis. Exactly right. Remember, the organism has two R's, but those R's separate during meiosis. So C should be your answer there for clicker question number 2. And do you remember we also touched on some non-Mendelian traits? So for instance, we looked at alleles that don't have a clear dominant and recessive version. One example was incomplete dominance, where the heterozygote has an intermediate phenotype to the homozygous. And another example is codominance, which we find in the ABO blood types. You should remember this for the exam. The ABO blood types are an example of codominance, where if an organism has the A antigen, they present the A antigen on the surface of their blood. If they have the B antigen, they present the B antigen on the surface of the blood. But if they have A and B, because they are codominant, both antigens appear on the surface of the blood cells. Whereas if they lack A and B antigens, they present neither on the surface of their blood cells. So you should remember that an example of codominance, an example of this non-Mendelian trait, is the ABO blood types. Next, You should know that pleiotrophy is when a single gene has many effects on different phenotypes. So, for instance, this gene that causes Marfan syndrome, this is a mutation in a gene encoding connective tissue, having effects in the eyes, the bones, the joints, the hearts, and the lungs. So, when a mutation in one gene affects multiple different organs or multiple different phenotypes, that's pleiotrophy. We also talked about pedigrees. Do you recall that pedigree analysis can be done on recessive genetic disorders as well as dominant? If a disease is inherited in a dominant fashion, then only one allele is required to show the disease. So let me ask you, let's see if you can beat Wicket. Can we have a dominant disorder, a dominant genetic disorder? Can you have carriers? Remember healthy carriers? Healthy carriers are people who have one diseased allele but don't show the disease. Can you have a healthy carrier when it comes to a autosomal dominant disorder? Can you beat Wicket? That's right, Wicket. You can't. So, in a dominant disorder, a dominant genetic disorder, there are no carriers. If you have even one copy of that diseased allele, you're going to show the disease. Does that make sense? However, in a recessive allele or recessively inherited genetic disorder, you can have healthy carriers. These are people with one functional copy of the gene and one diseased allele for the gene. These are called healthy carriers. So let's answer clicker question number 5 together. This pedigree tracks an autosomal dominant disorder. What is the genotype of 1 2? Let's talk about 1 2. So 1 meaning the parents, and 2 meaning the father. This is the father. What is the genotype of 1 2? So if this parent was homozygous, then every single offspring would have the disease. But does every single offspring have the disease, or is number 3 an offspring without the disease? Number 3 does not have the disease. So that tells me that this parent, the father, actually does not have 2 diseased alleles. He has one diseased allele and one normal allele or wild type allele. So he should be B, heterozygous. If he was homozygous, all of the offspring would show the disease. Does that make sense? I hope it does. And then lastly, you should know this for sure for the exam, and that is that some traits depend on multiple genes. So, for instance, height, skin color, these are types of traits that require multiple genes, that depend on multiple different genes. This is known as a polygenic trait. So, you should know that height and skin color are polygenic. They don't depend on one gene. They rely on multiple different genes. And that leads to the end of chapter 10 and the end of exam 4 review. I hope this all made sense. I wish you well on exam 4. Do your best, and I'll see you guys for exam 5 review next.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.